Well, hello, this is Miss Dagenford, and we are starting our unit on protein synthesis. And so I wanted to give you a little overview of protein synthesis. You should remember from your intro bio days some of the information you're going to learn, and it probably will come back to you as we're talking about it, as I'm presenting it. So we have talked so far this year about proteins. We mentioned a lot about proteins that are, we talked about membrane proteins that are receptors, and we talked about enzymes that have a very unique shape. We talked about all of these specific different proteins. And we also just finished DNA. And we remember that there are very important functions of DNA. It holds information, genetic information. And so the connection between these two is quite profound and very, very fundamental to understanding um, how cells work and how biology works. And so a lot of this information started in the 60s as we were starting to make connections between what does DNA stand for, and what's the connection between DNA? So one of the first things we understood is that a gene can code for a protein or polypeptide product. Now we've since expanded it, but one of our first understandings is that of the central dogma. And in the central dogma, we start with DNA, and of course DNA, that would be a gene, and we go through and we then copy it to make mRNA, and then mRNA is used to build a protein. And very early on, that was coded as the central dogma. And here you can see in the two pictures from our book um, the two representations. Now, one of these cells is eukaryotic, and one of these cells is prokaryotic. And I'm going to go ahead and number them just because I'm going to be asking you a question in just a sec. I would like you to be able to tell me, just as a quick review, which one is eukaryotic, number one or number two. And hopefully, if you recall, the picture number two would be eukaryotic, and hopefully you remember that that's because it has a nucleus, ding, ding, ding. And then we have a prokaryotic cell up here. Now, um, here's our, of course, cell membrane around our cell, and in a prokaryotic cell without a nucleus, the DNA would be found just inside the cytosol, the cytoplasm. And so here we have our DNA strand, double helix, no problem there. And notice we have our DNA, and as we follow the arrows, here would be messenger RNA, and we'll talk about its function. This video is just to give you a brief introduction, a layout of the land, so to speak. And then down here would be a ribosome, the brown, and the ribosome is going to be creating a polypeptide or protein. Um, and the polypeptide is going to be made of amino acids. And so the processes, and you should be able to tell these apart, you should know them. The process of taking DNA and copying the information by making it a messenger RNA, that would be transcription. And I like to remember that somebody who is just transcribing, or a scribe, or a prescription, well, you're writing something. So DNA and mRNA, you're copying, you're writing down the message. Now, going from the mRNA to a polypeptide, you're building something. So I like to think you're translating the message. So translation is that process by which we go from the messenger RNA and use that to build our polypeptide or protein. So that is kind of the central dogma in a prokaryotic cell. Now, again, we've since expanded. It's not just mRNA. We'll talk about that. So here's our eukaryotic cell, and hopefully there are some similarities. So, for instance, I don't think that you should have a problem with this. This is DNA. And let me just go ahead and get some labels here, and then I'll pause the video and ask you to identify. So here we have step A, and now there's an extra thing in eukaryotic cells called processing. This is our RNA processing. And the RNA processing involves modifying the mRNA strand. And then I'm going to label down here. And you should be able to tell me what is that step from DNA to mRNA. And then you should be able to tell me the step from mRNA to a polypeptide. Now, notice the difference. DNA is inside a nucleus now, so would be the process of copying it to an mRNA strand. And then the mRNA would leave the nucleus to go to the cytoplasm, whereupon it's going to be used to build a protein. So there's our step two. There's our cell 2, or eukaryotic cell. Now, when we look at the basics, one thing that we need to understand is in an entire DNA strand, you might have 3 different, well, more than that. But in this picture, we're representing 3 different genes. Just for the sake of argument, let's say that gene 1 is the gene for ATP synthase. By the time you're done, you would have our lovely little mushroom ATP synthase that is building our ATP molecules. Let's say gene 2 would be our catalase enzyme gene, and then gene 3 could be a gene for microtubules. Now, this is not to say they're all on the same DNA strand. They could be on a different chromosome, but let's just use those for examples. Now, in our DNA strands, you have, we know, 2 sides. We have 2 strands, the double helix. And you should recall our 3', 5', and of course the anti-parallel nature 5' to 3'. Now, one side is going to be called the coding strand, and one side is going to be called the template strand. So let me clean this up a little bit. So notice in our mRNA strands, now mRNA, there's 3 differences. We have, notice U for uracil. So RNA, off to the side here, RNA has uracil, and uracil replaces thymine. And then the other part that gets replaced is the sugar. Now, you can't see it on this diagram, but RNA has ribose instead of deoxyribose. And then RNA is single-stranded. Now, that does have some interesting characteristics. Because it is single-stranded, RNA can fold back onto itself. Now, you'll notice that in this diagram, and you got to kind of keep everything straight. This top strand is what is used to build the RNA strand. Because notice A would match, or complementary match, U, C would match to G, and then here we have another G for another C. So the strand that is used to build our RNA, that's called the template strand. Just like in art class, you would use a template to build a shape. Well, this DNA strand is what you're going to use to build your mRNA strand. Now, what we have broadened our understanding to include is that DNA can be transcribed to also make other kinds of RNA. Transfer RNA, ribosomal RNA, and so it doesn't have to be an mRNA strand. But for the purpose of protein synthesis, we'll focus on that for now. So we're going to transcribe our DNA into mRNA, and then we're going to use that mRNA strand to build our amino acids, and that would be translation. And we're going to get a polypeptide. Now, here's where we're hopefully on familiar ground. We've got a series of amino acids. And how this works, and this is where, again, you're probably feeling somewhat familiar because you probably remember this from your biology days. What we do is we take a triplet of nucleotides, and that triplet stands for an amino acid. It's used as a representation. We know that UUU is the codon, and that's what we call when we have a triplet. A codon would represent one amino acid, and a codon is made of 3 nucleotides. And the scientists broke the code back in all throughout like the 60s. We were kind of working up the code. Very painstaking set of experiments that we'll go over and kind of understand how they did it. It's rather wild, very, very wonderfully done experiments. And so what we probably remember is using this chart. And this chart, of course, in this one, you're just seeing the abbreviation. But if you recall the way it works, you have the first nucleotide of the codon over on the left, and then you have a second nucleotide on the top row, and then all of these are the third nucleotide. So if let's go with UUU, well, you start U, and that means that it's anywhere in this row. And then the second letter, U, and that you go down. And so now it's this little square. And then the third letter, U, you follow over, and that's our phenylalanine. Now, you would never need to memorize this chart. You'll probably start to know a couple of them because I like to use some that I know. And so you might recall, and we did this with our monster lab and things of that nature. So just let's practice AUG and AUG. So we're going to start A. There's our first letter. U, second letter. So we go down for that. And then G is our third letter. Go over. And that's our start codon, start methionine. All polypeptides have kind of that first codon. I shouldn't say all. There are some weird exceptions, but that's pretty familiar. And then CCC, so C, first letter, C, second letter, and then here's our third letter, C. We go over, and there it is. That stands for proline. And then UAU, UAU, tyrosine. And then UAG, yay, stop codon. And we'll talk about the significance of that. How does that work? But right now we just want to familiarize ourselves with kind of the basics here. And then the next video we'll talk about the details. But that's just a rundown, and that should help you get started with this unit. All right, see you on Monday.